你俩的对顾都是从在大会院登记的。对，大会院登记。对，大会院登记，然后后面是您打电话，后面有是。你先改一下，改。我直接改到。大会院登记。
好，会场听得见我声音吗？会场听得见我声音吗
Hello, can you hear my voice?
？Hello, can you hear me?
Yes, Funchen, I can hear you perfectly well.
Okay,
So,
thank you.
Shall we start?
Yeah.
We have everybody.
Yes. 非常感谢啊，大家早晨好，非常感谢。
Good morning. Thank you very much for participating in this break session B of the CIFSQ and Rapid Microbial Detection Sample Preparation. I'm Li Fengqin, research scientist from the China National Center for Food Safety Risk Assessment. I'm very honored to join Seamus Fanning from University of College Dublin. And we are very grateful for Professor Fanning for his participation because it is midnight in Ireland. Thank you very much.
Let's get started. Thanks very much for coming to a rapid microbial detection and sample preparation session at China International Food Safety Conference today. I'm Li Fengqin from China National Center for Food Safety Risk Assessment. It is a great pleasure for me to co-host this conference with Seamus Fanning from University College Dublin. Due to the time difference, it's midnight in Ireland now. Thanks very much for Professor Fanning's hard work. Firstly, I would like to invite Professor Fanning to host this meeting first. Are you online?
I'm online.
Please.
Yep, I'm online, Fengqin. Thank you very much. And it's a pleasure for me to attend the meeting once more. I'm sorry that I'm not there in person, but it's always a pleasure to come back to China whenever the opportunity presents itself to visit my old friends like Fengqin Li. So thank you, Fengqin, for this. And without further delay, let me introduce the first paper, which also comes from Ireland this morning. So we are up very late. And it comes from my good friend and colleague, Dr. Guerrino Macori. So Guerrino, just to give you some background, is a research fellow in my lab, and we're very lucky to have him. He's an expert in DNA sequencing, and he's going to tell us today about the use of next generation sequencing to improve food safety and quality in different aspects of the food chain. So Guerrino, without further delay, over to you for your presentation, please. Thank you.
Yes. Hello, everyone, and thank you very much, Professor Fanning, for the kind introduction, and good morning to all the colleagues at the conference. My name is Guerrino Macori. I'm a research scientist, a molecular microbiologist as a background, and bioinformatician in UCD at the School of Public Health, Physiotherapy and Sports Science. I'm very pleased to be here today with all these fantastic speakers in this session as well, representing the Center for Food Safety, the University College Dublin, Ireland. The center is located on campus at the O'Brien Center for Science, and CFS, Center for Food Safety, is part of the Institute of Food and Health, and is well recognized as an academic center of excellence in food safety research, directed by Professor Seamus Fanning. Today, I'm going to present a few projects I followed on the use of next generation sequencing to improve food safety and quality. And as you know, the advances that this technology have contributed to the revolution of food safety, facilitating in some sort the detection and characterization of foodborne pathogens, using, for instance, whole genome sequencing as a tool for genotyping microorganisms, and the crucial role as well for highlighting important features such as antimicrobial resistance and for discovering potential virulence factors. And then the use of new approaches and models for study, for instance, complex microbial communities in food, applied both in food safety context, food quality, and to reveal traceability and authenticity. I will try to go through this journey today with you. So whole genome sequencing provides a comprehensive view of the genome structure and gene content of microorganisms, and is now gradually replacing well-recognized standardized protocols like the PulseNet for its higher sensitivity, specificity, and interlaboratory comparability, as well as timely resolution when compared with traditional molecular methods such as the panel of virulence factors that you can see in the slide at the center. this whole genome sequencing in general has been applied for genomic comparison of microorganisms that are associated mostly with foodborne outbreaks. And the example of an investigation is well depicted here in the left-hand side of the slides, and you can see that has been used for surveillance of high burden diseases for almost a decade so far, allowing the identification of pathogens in the foods responsible for the infections. A typical approach of a whole genome sequencing is summarizing the scheme here, where we have an upstream preparation. So you can find all the protocols for the sample analysis according to the presence of reference sequences, and also a downstream process where we prepare the results and present to stakeholders or clinicians and other researchers. One of the most common foodborne related diseases is the staphylococcal food poisoning, resulting from the consumption of foods containing sufficient amounts of staphylococcal enterotoxins. So the staphylococcal food poisoning is associated with enterotoxigenic Staphylococcus aureus strains that may produce one or more heat-stable enterotoxins in food. And these will cause then vomiting and abdominal pain as well in humans. And considering the high percentage of all European outbreaks reported are caused by staphylococcal enterotoxins, is very important implementing surveillance plans, and also provide a sort of reference genetic material that could be used for official controls to comply with regulation and validation studies, for example, ring trials, and have access to a very well characterized and stable reference strain might be fundamental for assuring the quality, which unfortunately there are several variance factors, including this test, that correspond to different genetic profiles of strains. And in this study, we characterize 6 strains. These were very particular for the context of toxins that we're able to produce, and were then also provided to laboratories that are involved in official controls for food safety state laboratories. A collection of strains were tested for stability, and in collaboration with the European Reference Laboratory for coagulase positive staphylococci, the 6 profiles shown in this table were included. And as you can see, all of them were isolated in vivo, either because responsible for foodborne outbreak or in the context of food control plans of these reference laboratories. The strains were characterized also through molecular tests for the presence of 11 enterotoxins that have the feature to be encoded in mobile elements like plasmids, prophages, or specific pathogenicity islands and gene clusters in the chromosome. And interestingly, the 6 strains were all toxin producers in vitro. This was then completing providing a nice collection of factors that might be used then to compare with other bacteria responsible for foodborne outbreak. We were also interested in providing a database very well curated with all the possible features of these strains, in particular the available staphylococcal enterotoxins, including the variants, and the ones for which is not possible the detection for diagnostic purposes in matrices as foods, or because there are not methods available for their detection just yet. And we did the same for other databases. For instance, some of them were collecting specific sequences for pathogenetic islands, and also a well-curated collection of more than 4,000 nucleotide sequences that have described in Staphylococcus aureus plasmids, that revealed also the presence of 3 enterotoxin genes in plasmids not well described before. Now, the powerful aspect of generating whole genome sequences is to provide sets and collection that are rich of metadata that can be then mined, and also to link cases and hosts to specific genotypes, for instance, and phenotypes. And this eventually will be used in the future for training machine learning models for source attribution. Now, this is very interesting from the food safety perspective, especially for food safety, for laboratory involved in food safety control, food safety and controls. Because if we can, for instance, we use this approach for revealing even hidden sophistications. For example, there are many other enterotoxins produced by Staphylococcus aureus, and the different genetic variants have been described with a specificity of the host. I mentioned here in this slide, you can see a sheep, a cow, and in fact, we were able to find toxins that were produced by a strain with a specific variant of a C type that is specific again for sheep. And this was very interesting because actually the milk that we were testing was supposed to be only cow milk. And then we found out that there was a sophistication in terms of contamination of the milk with sheep milk. In this study, we also studied, checked the rinds and the cores of all the cheese of a batch of natural contaminated cheese made from raw cow milks. We investigated the Staphylococcus aureus presence and also the toxins in a different area of the cheese. We detected several strains. And again, what was very interesting to find out that one of the strain and the profile of this strain was that they had origin as a ovine. So it was actually from milk that was not supposed to be there. So using well-characterized strains as reference material in combination with this database that I just mentioned, we offer then a comprehensive portrait of the strains that we can test and we can have and isolate in the laboratory. And then this, of course, will describe better the bacteria that we are testing and they're relevant for food safety. Another approach, an experiment I wanted to include in this presentation is the usage of metagenomics and the different flavors that can be applied to improve food safety and quality. A microbiota, just as a definition, is the community of microorganisms living together in a particular habitat, and a microbiome is described as the complex microsystems of interlinked networks and functions. This includes microorganisms and their genes. So we can study and describe composition of bacteria in food, sequencing the ribosomal RNA gene, and give information also on the diversity of the population present in a sample, such as the difference within a sample in the form of alpha diversity, and also the beta diversity, which is the difference between the samples. Now, we can compare the sequence that we obtain from samples that we are sequencing for the presence of 16S RNA genes. And this is called amplicon sequencing approach. And this describes the abundance of taxonomic units, as you can see in the table. So we have an abundance for every single OTU here in the column. And we did this to evaluate the effect on the microbiota by changing conditions for preparing this product. This product is called lardo, which is a type of salumi made by curing strips of fat back with rosemary and other herbs and spices. So we tested 3 different products. One was the artisanal and traditional production with the use of 30% of salt brine and then cured at 8 degrees. The other 2 productions consisted in a variation in terms of salt content and also temperature of storage. And one was cured at 2 degrees and the salt content was 10%, and the other one was had a salt concentration of 2.5 and was maintained at 4 degrees. The main taxa identified by sequencing are reported here. And you can see as well that in the graph in the right-hand side, with the red circle represents the indexes of the PCA graphs, of the production, which is artisanal and traditional. In the left-hand side, you can see in blue and green the new production, the novel approaches. And interestingly, from the food perspective, these two productions were characterized by the higher presence of taxa recognized as not desired in food processing environments. For instance, we found different bacteria that are actually spoilage agent, and this, of course, is not acceptable when we have a production that must go in the market. And with this approach, we were able to describe the safety of this product, but also that unfortunately we cannot really change, even though we need to go and help the consumers to reduce, for instance, salt content, but this changing drastically changes as well the formulation in terms of microbiota, and might also give access to pathogens to start to grow, or spoilage as well, pathogen that can grow and then damage your batch of production. Now, metagenomics can be used also for describing the microbiome of fermented fruit products. And in this study, we tested two different batches of fermented sausages. One was inoculated with a commercial starter culture composed by Lactobacillus sakei and the Staphylococcus xylosus, and the other was a spontaneous fermentation. We sampled at different days and analyzed as well the meat with a shotgun metagenomic approach. And also we tested the volatilome profiles in order to have a look at, with gas chromatography, to the presence of chemicals that were maybe interesting from the commercial point of view for the flavors they were producing. And we did as well a panel liking test in order to see as well the consumers, how they would appreciate or not these two different batches. So the results were very clear in terms of diversity of the population in the spontaneous versus the inoculated batch. And you can see in the left-hand side a huge diversity in the spontaneous, indicated with S, and the inoculated in the right-hand side. While the volatilome profiles shown a very peculiar patterns for the spontaneous fermentation, indicated with the letter S in this heat map in the lines that represent the different volatilome compounds. Every single line is one different volatilome compound. And you can see that those are the spontaneous, and we have the row that are lighted in with the molecule identified. Then the metabolic networks reveal the relationships between the metabolisms of carbohydrates, which are in red in this graph here. And we have amino acids in yellow and lipids blue, with the spontaneous, which are the larger spheres in cyan, and the green represents the inoculated batches. And you can see as well the metabolic pathways and samples are connected with lines for which the thickness is proportional to the abundance of that pathway in that connected sample. For instance, the inoculated had a strong connection with pentose phosphate pathways and glycolysis, in addition to starch and fructose metabolism, especially in the first step. So the I3 that you can see in the middle is actually the inoculated batch at 3 days of the starting process. There was in fact, abundant after 3 days of fermentation. On the other hand, the same pathways in the spontaneous samples, so the S samples, were more abundant after 7 days of fermentation, most likely due to the fact that the evolution of the microbiota was delayed because of the hard time that these microorganisms were having to survive and to start to go for that metabolic pathways. So significant differences were found as well in the liking of samples based on flavors and odors, suggesting a higher preference for the consumers for the spontaneous fermentation samples, while the inoculated samples exhibited the lowest scores in this graph, and most likely for the presence of high concentration of acetic acid, where the descriptor were pungent, acidic, cheesy, and vinegary for what concerned the batch that was actually inoculated with the starter community. This shown how starter culture can significantly affect the development of the microbiota, as well as the gene content and the volatilome profile. And the strong connection is valid between the starter that develop during fermented sausages ripening and the sensory characteristics of the final product. However, the risk, because we did as well microbiology analysis with culture-dependent techniques, so we found that actually contamination with pathogens on the spontaneous fermentation, and this, of course, is not really acceptable from the food safety point of view. The last project for the last couple of slides that I want to present today is a characterization of microbiome of milk filters. These were sampled for evaluating the resistome, which is the full set of antimicrobial resistant genes. But we also tried to reconstruct full genomes from the metagenomic data. And we were able to assemble actually 8 high-quality metagenome assembled genomes that were furtherly characterized. And we obtained even a more complete picture of the gene, conferring resistance and their relationship with the genus identified through metagenomic analysis. So the raw milk can be considered, according to this approach, a source of AMR bacteria, so bacteria that have antimicrobial resistant genes. And the reason, and these, of course, highlight the importance to properly inform food businesses, operators about the risk associated with poor hygiene practices in the dairy production environment, and also consumers of the potential microbial food safety risks derived from raw milk products consumption. And of course, this will nicely translate how is important to have new technology, but at the same time as well translate this information in risk assessment outputs that will be then used in the future as a next generation of food safety controls, which will, in my opinion, would include also the evaluation of high quality MAGs, these metagenome assembled genomes, as a tool for precision food safety. And with this last thought, I want to thank you again for the kind attention and invite to be part of this session, and all the people involved in the studies I presented today. So there were so many, and in particular, I want to thank the director of the Center for Food Safety, Professor Fanning, and also the colleagues that work in the team, and many collaborators in different projects. And I'm really happy to answer 20 questions with my.
So thank you, Carino. And I think Funchin is going to take the questions.
OK. 大家有什么问题吗？对刚才 Grano
Any questions from the floor? If you have any questions, our professor will be very happy to take your questions.
Do you have any questions?
Funchin, there's one question in the chat here from Yingping Dong. So she asks Carino, can you get better results if you do metagenomic sequencing of a clean sample, such as a factory environment sample? And is it okay to increase the collection area? So I think what Dong is talking about is taking a sample, an environmental sample, and how big an area, and can you get a pretty clean DNA extraction for that to generate some sequencing?
It's a very good question, and like my understanding in terms of increasing the numbers of samples, for instance, and the pool, it doesn't always give you a very good output in terms of quality of DNA that you are extracting. So if I understood the question, if you have a bigger samples, of course, you're increasing the amount of DNA, but this doesn't mean that you're going to have a good quality DNA anyway. I don't know if this was the question. Of course, it's very important as well to test your methods all the time in order to be sure that you are actually able to produce anyway DNA that is a good quality. For instance, if you have a sample that you want to test with a new kit of extraction, for instance, or a new technology, I always suggest to the student and the colleagues that we have in our center to test always a mock community, which is a community of microorganisms that you can use to spike your clean sample, your blank samples, and then do your process of extraction and sequencing in order to see if you actually are able then to find out those communities that were composing, that were part of the pool that you used to spike it.
Okay.
Janelle, and back to you.
Okay.
We have a question here in Shanghai, a great question. So my name is Guangtao Zhang from the Mars Global Food Safety Center. I have a question for the presenter, which is, what is the regulatory environment on using metagenomics for food integrity? For example, in this case, for cheese, or maybe for other type of meat. Yeah, so is there any suggestions, or is there any kind of forward outlook from you or from the presenters, either, or from the chairs?
Yeah, maybe I can start a conversation and yeah, maybe I can start the conversation, then we can open, I suppose, comments from the other chairs that I suppose that they have a lot of insight on these as well. So my suggestion is there's not, first of all, there's not a regulation on terms of metagenomics in terms of which type of technology you need to use to be sure that is going to be a good output, or also, for instance, having reference materials that you can use for doing this test and so on. And this, of course, is a big gap in terms of regulation and quality in the lab, if it makes sense, because then all the strong part in terms of validation is on the final user. So it's the laboratory that needs to validate their own methods. Of course, there are guidelines, and in particular in Europe, European Food Safety Authority has prepared different recommendations on this part, and there are also independent groups are working in the ISO for generating novel guidelines as well, that I hope that they will come out soon with some solution in these regards. The big issue is also technological, because every single week I see a different update in terms of even hardware in this sequencer. So we have a new technology every single week. This means that it's very hard to validate a method that then after a week is not actual and current anymore.
Thank you very much.
Okay, time is limited. We may move to the next presentation. Is okay, Fanny?
Okay, Chongqing, over to you.
Okay, thank you. Thank you very much.
Thank you very much.
拜拜。那么我们第二个演讲者呢是来自
Our next guest speaker is from Mars. Mr. Ge Chongtao, Senior Research Manager, Microbial, Mars Global Food Safety Center. He graduated from a prestigious US university and got PhD degree. And he works as a leader for the Microbial Center of Mars Global Food Safety Center, responsible for the rapid detection.
He received his PhD in food science and technology from the Ohio State University with a focus on food microbiology. He leads to a microbial risk management research program in the Mars Global Food Safety Center, which is focused on the development of foodborne pathogen rapid detection and inactivation. Please, Mr. Ge.
Yeah, thank you, Dr. Li, and really glad to be here to present some of our work in the Mars Global Food Safety Center. And today I'm going to talk about evaluation of Oxford nanopore sequencing for Salmonella genome comparisons, which is a really good tool for contamination source tracking of Salmonella in the industry. So please allow me to introduce Mars before the study. So Mars was actually founded in 1911 in the US. So Mars has kind of over 100 years history so far, and the headquarters is in McLean, Virginia, in the US. And so far we have over 140,000 associates around the world. And last year, our net sales is about 45 billion. And you may not kind of necessarily know about Mars, but you definitely know about some of our brands. For example, the sneakers, Dove and Wrigley Extra, which is in our confectionery business. And also we have Pedigree, Whiskas, Rocanin, which is in our pet care business. And also we have some other types of food lies in the Mars food business and Mars Edge business, focusing on human nutrition. So with the globalization of the food supply chain, actually we saw the quality and food safety has a domino effect. And whenever there's a food safety or quality issues, it will kind of impact on the download manufacturing and also consumers, and then impact the whole society. With that, actually, we found the Mars Global Food Safety Center in 2015 in Beijing, China, and to collaborate with internal and external stakeholders to work on some focus area in the food safety to increase the food safety bar of the whole world. And this is the Mars Global Food Safety Center approach. So actually we establish internal and external research projects and collaborating with a lot of top universities, NGOs, and government. And also we built the training program, which is not only for the Mars associates, but for the whole industry peers and also government and NGOs as well. And with the whole partnership and collaboration, as I just mentioned, that we'd like to show the leadership of Mars in the food safety area and to increase the food safety bar with all these stakeholders globally. And here presents the three major areas in our research programs. So the first one is mycotoxin risk management. We are working on a lot of projects to prevent the aflatoxin genesis, and also work on come up with a better solution for managing mycotoxin risks, and also repurposing the raw materials. And in the microbial risk management, which I'm working in, so actually we are working on using whole genome sequencing methods for pathogen detection, confirmation, and source tracking. And also we're gonna use some of the metagenomics tools to monitor the microbial ecology in the environment to predict the risk of contamination. And the third area is called food integrity. So in this area, we have a variety of directions, for example, to develop some tools and standards of analytical methods for food hazard detection, and also develop some methods on real-time surveillance, and at the same time work on the inline and outline testing in our manufacture plants. And so today, I'm going to talk about the application of whole genome sequencing in the food safety area. And So as you may know, with the kind of COVID-19, a lot of people, they're already kind of familiar with this PCR test, and also whole genome sequencing for source tracking regarding the COVID-19 globally. So actually, since decades ago, actually, the whole genome sequencing has been applied or tested in the food safety area. Like for example, in foodborne pathogen surveillance, foodborne pathogen source tracking, which I'm gonna introduce a little bit, and the foodborne pathogen risk assessment, and the food production microorganism risk and safety analysis. For example, the lactic acid bacteria to make yogurt, etc. And also, besides the microbiological area, is also being applied in the food fraud and authenticity analysis. And around the world, we have seen that whole genome sequencing platform has been established in a lot of countries, like the US, and also in China and in EU. So actually, FAO has developed a program that targeting to help the developing countries to build their whole genome sequencing platform for the food safety aims. So we already kind of knew that whole genome sequencing is a really good tool in terms of in the incident analysis. And the question is that how we can apply this technology in the industry. Given the kind of a whole genome sequencing platform, and it requires kind of high skills in terms of technology and also cost of testing, and the space in terms of the labs. So sometimes it's really hard to deploy this method in the plant lab. And in about like 2017, we found that there's a emerging sequencing technology called Oxford nanopore technologies, and they actually can provide a portable device as well as a benchtop sequencer. So actually, we were just thinking about if this device, if it's kind of feasible to feed our targets, probably we can widely used whole genome sequencing in industry, in our lab network for source tracking. So Oxford nanopore technology actually is considered as a third generation sequencing technology. Not only they can provide a portable device, but also it can generate real-time data, which means you don't really have to wait until the sequencing is done. You can just extract the data anytime while the sequencing procedure. And on the left side actually shows the mechanism of the sequencing. And when the DNA goes through the nanopore, driving by the motor protein, and it can read, it can kind of change the electric signal. And then there's a software inside that can translate the electric signal into the gene signal, which is ATCG, based on the difference of the electric signal, and then you can get the whole sequence of the target genome. And the question is that if it's feasible, and how the kind of how it performs in terms of to fit our business need. Like for example, and we start with Salmonella serotyping, which is an important procedure in contamination investigation. We all knew that Salmonella has over 2,500 different serotypes. And if we do traditional like serotyping using cough-and white methods, it always generates wrong data. And also the turnaround time is about 1 week. So that's why a lot of regulators, they tend to use whole genome sequencing for the serotyping. And actually we started this experiment in 2017. So we compared the ONT. We compared the serotyping in silico serotyping using ONT versus using Illumina, which is considered as a golden method currently. And we proved that with using ONT sequencing, we're able to get exactly same serotype as use Illumina sequencing, and the whole sequence can be finished within 1 day from isolate on the plate agar until you get the real name of the serotype. And later on, because the serotyping is the first step, and to be able to get the real source tracking, you really need to use some like downstream bioinformatics analysis, for example, SNPs or whole genome or core genome MST, to find out where is the true contamination source. So with that, actually, we collaborated with the University of Georgia and also Cornell University, and to select a total of 50 Salmonella enteritidis isolates from US CDC and isolated from 18 different outbreaks across the US in the past 20 years. And then we do ONT sequencing and Illumina sequencing in parallel, and using Illumina sequencing as a reference method to see how ONT sequencing works. And then we do bioinformatics analysis to perform the SNP, CGMST, and then use some stack analysis to compare. And our success criteria is that the pattern of subtyping generated from genome comparison methods like CIFSQ and SNP or CGMST for Salmonella contaminated source tracking using the two technology, especially ONT sequencing, is comparable with using Illumina sequencing. So here is a entire workflow, and with ONT we use a GridION, which is a benchmark sequencing, and also their latest super accuracy based calling software. And so for Illumina, we use the next sequencing, NextSeq to do the Illumina sequencing. And for CIFSQ SNP, actually is use a raw rays as the input, and then come up with the CIFSQ SNP matrix and the trait, phylogenetic trace. And for CGMST, actually is require assembly. And for this, actually, we first, in our preliminary experiment, we found that because of the homopolymer errors generated in the whole in ONT sequencing, it's really kind of affect the indels, which significantly impact the downstream CGMST analysis. So that's why our partner in University of Georgia, they come up with a procedure called homopolymer error correction, and then to be able to get more accurate CGMST results. So this picture shows the SNP analysis, the comparison, as you can see on this picture. And on left side is the Illumina sequencing, and on the right side is ONT sequencing. And we can get exactly same clusters by using the two methods. And all the isolates they cluster by the outbreaks, and the relative position of the branches are same between the traits built by Illumina and ONT sequences. And we also compare the SNP difference between Illumina sequencing and ONT sequencing. We found that among the 50 isolates, so only 3 of them actually has one SNP difference between Illumina, ONT, and all the others. They don't have any SNP difference. And we also combine all the ONT and the Illumina sequence in one phylogenetic tree. And same as the previous slides, actually they cluster by outbreaks, and all the ONT sequence, they cluster together with their corresponding Illumina sequence as well, which means ONT sequencing is a promised tool to perform SNP analysis. And we did some statistic analysis. And on the left side, there's a pairwise distance comparison of the pairwise distance by ONT race and Illumina race. And when you look at the formula, actually, we found that the intercept is really close to 0, and R square is equals to 1, which means it's quite promising. And there's an ONT sequence and an Illumina sequence is highly highly similar in terms of conducting SNP analysis. And also we analyzed the sequence depth to and want to find out which is a kind of threshold that we can generate the qualified SNP analysis. And with that, actually, we found that 50 when the sequence depth reach to 50x, and we can call out around 99.3% of the SNPs. And there's only kind of needs 1.7 to 3.2 hours of sequencing. In average is around 2.3 hours of ONT sequencing. And also we perform the CGMLT and come up with this minimal spanning tree. And so here we show that different sequencing different types of sequencing data, Illumina data, ONT data, and ONT data after homopolymer error correction, and also hybrid by combining the Illumina data and the ONT data. We took the hybrid as a ground truth and as a reference to compare other different types of the sequencing data. And we found that without homopolymer error correction, and it may generate inaccurate cluster, and they might not necessarily cluster together by using only ONT sequencing without any correction. So, which means the homopolymer error correction is really, really helpful. And also, as we can see here, so without the homopolymer error correction, the average allelic error is around 16, and after the correction actually is lower to about like 5 allelic errors. And with the homopolymer error correction, we are able to generate the qualified CGMLT data, as you can see here. With the comparison between Illumina and ONT, there's no much difference. They also kind of cluster, all the isolates clustered by outbreaks, and the relative position of all the clusters, they are exactly same between Illumina and ONT minimal spanning tree. And when we look into the statistical analysis between use Illumina and the ONT sequencing to perform CGMLT, we found that with a homopolymer correction, so actually the difference between Illumina and ONT can be significantly decreased. And samely, we analyzed how the sequencing depths affect the accuracy of CGMLT. We found that when the sequencing depth is around 100, and the allelic errors significantly decreased. And also, so we also kind of calculate the sequencing time to reach to 100x steps, and it's around 3.2 to 6.6 hours of sequencing. And here we got the conclusions. The first is a genome comparison, and we in this study, SNP and CGMLT generated by ONT sequences using super accurate base caller are comparable with Illumina sequencing. And the homopolymer error correction step enables the use of accurate CGMLT analysis, and also by multiplexing 3 isolate per flow cell, sufficient sequencing depths could be obtained within 7 hours in terms of performing SNP and CGMLT of sequencing for genome comparison analysis. And then we can figure out where is a high likelihood of the contamination source. And earlier we published this study in Applied and Environmental Microbiology. Feel free to check it out if you're interested. And also here we'd like to thank our partners, Oxford Nanopore Technologies, and also University of Georgia, Dr. Xiangyu Deng's lab, and Dr. Martin Whitman's lab in Conagra University. And with that, thank you for your listening, and feel free to let me know if you have any questions. Thank you.
Thank you very much. 非常感谢葛先生的精彩
Thank you
演
very
讲。
much for your excellent sharing. Any questions from the floor or online? Because later on we have a 10-question Q&A session, a 10-minute Q&A session. And I have discussed with our organizers, so we can allocate this 10 minutes to the end of each session. So any questions from the floor? For Mr. Go, you can raise your question now.
有问题吗？
李老师这边没有。
好嘞。
No question on site.
这样子的话，我是不是问一个问题？
So I'd like to ask you a question.
说你们马氏的话，我知道，因为是
Mars, as far as I know,
就是测序仪的，
you have a sequencer. So in the process, in the food processing,
他讲的那个就是
and also as we have heard about the metagenome sequencing from our last speaker. So you have metagenome sequencing process, right? And what problems have you dealt with by this approach? Thank you very much. It's a very good question. Our previous work focused on extracting the string and then to have analysis. So now we are considering to take the metagenome sequencing as our approach. So as we have heard from my fellow speaker, we should manage the extraction first, and it will save us lots of time in this process. But as we have seen, the main problem for this metagenome sequencing technology is whether its accuracy would be enough, whether it's enough for the following up analysis. For example, among its current analysis or analytical approaches, the mainstream would be on the genome level. But for us, we're more curious about its serotype or its genome level, whether these data are enough for our analytical approaches following up. Now they have this new approach. They will do enrichment first, to manage enrichment first, to get enough volume so that the volume would be enough for our follow-up analytics. In this regard, it will save us a lot of time. On the other hand, we would have the ideal output as well. So currently we're exploring these approaches in this regard. And next year, we would find it possible or feasible to carry out certain projects in this regard. Any other questions?
好，那如果没有问题的话，那我们谢谢葛先生的那个精彩分
So
享。
thank you again for your excellent sharing, Mr. Guo. And we will
好，谢谢。下一个演讲者
proceed the next speaker. Merieux, Nutrisciences, Mr. Chen. He is congested on the road, so we will proceed. Our next speaker is Madam Dong Yingping, Associate Research Fellow, China National Center for Food Risk Assessment. Very experienced in the microbial research and works as a leader for lots of China's state-level research and development projects. So today, we'll share with us about application of rapid detection technologies in surveillance of microbial.
From China National Center for Food Safety and Risk Assessment. Professor Dong works in China at China National Center for Food Safety and Risk Assessment, mainly focus on the detection and identification of foodborne pathogens as well as probiotics in recent years. She received a grant from a China National Science Foundation of China, 2 projects from Beijing National Nature Science Foundation, and a subject of key research and development plan from a Ministry of Science and Technology. Now, please, Professor Dong.
谢谢李主任的介绍。因为今天参会的主要
Thank you
是
very much, Madam Li, for your introduction. So today, most of our attendees are Chinese, so I will present to you in Chinese. So dear colleagues, experts, and audience, good morning. We're happy to be here to share with you about this topic. I'm Dong Yingping from Microbiology Laboratory. So this is about rapid detection technology and its application and surveillance of microbials in food. So brief introduction to our lab first. We have altogether 19 staff. Most of them are very young. And the academic background is very high. So we are a very young and dynamic team. So this is about our main business scope. This is very widespread scope of us. The food microbial sector can be divided into 6 main categories. So our business relates to all of them, including foodborne bacteria, foodborne virus, harmful fungi, and foodborne parasites, biotoxin, species identification. So this includes foodborne pathogenic bacteria, microorganisms for food industry. We also work for the R&D of the virus. We just published a list for the microorganisms and the bacteria that can be used for the infant and baby food. So before the introduction of the rapid detection technology, I'd like to walk you through about microbial detection techniques first. So I have briefly wrapped it up into 5 different categories. From this page, you can see that these techniques include the conventional techniques, also on the basis of MS, protein, DNA, and the immuno testing. We also have the animal test. So these are our main categories of technique. You can also see that in the microbial detection techniques, it seems very different from the others. This detection will not be okay on the basis of a single technique. In our daily operation and daily detection, we also need to combine different techniques to complete this task. So next, I will walk you through about rapid detection and its application in our daily operation. However, I want to note that traditional is also very important in our detection operation. So very easy, simple example to share with you. Emergency response incident. We need to do detection for the BONT test. And when we do this, we use a lot of different techniques, different combinations. As you can see, we have listed them. So altogether, we've used up to 10 different techniques.
Because during emergency testing, we only have short duration of time, so the time requirement is very high. Therefore, we used 10 methods in parallel, including some traditional phenotype testing for microorganisms. We also use gene technique for testing. For rapid testing, we use ELISA. We also use MS, as well as the gene sequencing. We combine all these methods and we completed the task. Going back to the rapid detection method, to start with, I would like to introduce the currently prevalent method in our lab. It is also an emerging method in the past 2 years. Such a detection method, and after fluorescence PCR, it is like a next generation method. Its principle is similar to fluorescence PCR. The difference is that it is based on the genetic amplification as well as immunological method. It is like a combination. So this method is called recombinase polymerase amplification, or RPA. It also combines the lateral flow test strip immunologically, so we can achieve rapid detection in the field. Because we only need the normal temperature amplification, and after 20 minutes, we can have the test result. For such, in the temperature of 39 to 38 degrees Celsius, depending on the equipment. In these circumstances, we only need a simple equipment to achieve amplification. And after amplification, we can have visualize the result. It means that we can actually see the result. So we can achieve the rapid detection in the field, and it has good performance. I also list all the advantages and applications of this rapid detection method. We mentioned that for RPA, we only need the constant temperature equipment, so it's the lowest requirement for the equipment. What's more, we only need some half an hour to have the result. Also, the pre-processing of the sample is quite simple. If it's a contaminated sample, we don't need very complex pre-processing. We only need like the boiled method to extract the DNA. In terms of accuracy and sensitivity, it is quite high. We have a comprehensive comparison. We found that for RPA technology, its sensitivity and accuracy is superior to fluorescent PCR in certain circumstances. Therefore, in our lab, this method is widely applied. In terms of its application, we can have genetic testing of botulinum toxin type A, B, and E. No matter in China and in the world, we depend on animal tests to have the subtype of botulinum, which is a limit for many labs. But with this new technology, from the contaminated sample, it is quite easy to do the subtyping of gene. We also apply it in the emetic toxin of Bacillus cereus and also the norovirus detection and identification of Salmonella. For the botulinum toxin detection, we have promoted this technology in many provinces in China, including Hebei, Tianjin, Shandong, etc. So this method is particularly suitable for the large batch sample and a quick screening in China. If we have a large sample, if we use traditional method, it is time-consuming and effort-consuming. The result may not be good. But with a rapid detection, we can have the result quite quickly. Based on our own technology, we also applied a patent. Now I would like to introduce the application of our technology. For instance, on RPA, we have the quick test kit for norovirus. It can not only be applied to our routine testing, it is also applied to the security of the Winter Olympics. And we also gained recognition from the Olympic Committee. And we also have the genetic testing of BONT encoding genes A, B, and E. It shortens the time needed for the detection of botulinum. In the past, we needed some 1 or 2 weeks, but now with the rapid detection, we can shorten the duration of time to 1 to 2 days. For Salmonella, we also have invented an intelligent card reading system. We not only visualize the result, we also have the intelligent reading equipment for large batch of samples and testing. We can have intelligent reading of results. Therefore, no manual effort is needed. Apart from RPA technology, another quick detection technology is the prediction model technique based on machine learning. Now the popular concept is AI, as we all know. Through AI, we know that machine learning will replace humans to solve many problems. Of course, we need to have the standardized database. Namely, we tell the machine what to learn and how to learn. And based on the algorithms of the machine, and they can solve problems for us. I will introduce in detail to you about the application of machine learning in our lab. The first application is a machine learning-based intelligent identification technology. It is used in the MS technology. Another application is the algorithm-based intelligent identification. It is mainly used for intelligent analysis of genome data. Application 1 is the isolation and typing of Bacillus cereus from vomit of patients. It is based on the mass spectrometry because it is quite prevalent in labs. When we use MS for identification of bacteria, the result may not be satisfactory. They only tell us what bacteria it is. But we want to know through MS testing whether the strain has certain features. For instance, for the prediction of the phenotype of Bacillus cereus, we have our self-established database. In the database, we can tell the difference between the normal Bacillus cereus from the specific bacteria we want to pay attention to. For instance, the Bacillus cereus with the gene of vomiting. And after building this database through self-learning, the machine will tell the difference between the subtypes of Bacillus cereus. After building this database, no matter in our routine monitoring or some emergency diagnosis, we can tell the difference. For the 13 strains of Bacillus cereus from the vomit of patients, for the MS of this of these isolates, we compare it with our own database. We found that the 12 out of the 13 isolates are Bacillus cereus from vomit of patients. And we also have the PCR confirmation of these 12 isolates. And we confirmed that all these bacteria contained the gene of vomiting. It means that through the machine learning and we built the database of MS, it is 100% accuracy. At the same time, we compare the self-built database and database of MS for the 13 isolates. We compare the results between the two databases. We found that the database of MS only gave unsatisfactory results. They only tell us whether they have the genes of vomiting or not. Through machine learning, we can reach our target. It means that we not only know what bacteria it is, we also can find out whether it contains certain features. The second application is a prediction of drug-resistant phenotypes of foodborne S. aureus. We also use machine learning so that we can predict the features of genomes. For instance, we use the algorithms and we also use machine learning for the S. aureus. We have generated 2,000 chimeras. And we also correlated with the drug-resistant phenotypes of 11 antibiotics. So we'll know that which phenotype is sensitive and which is drug-resistant. Through machine learning, we can tell the difference between sensitive and drug-resistant strains. After this process, we give it a new isolate. The machine can tell whether the isolate is sensitive or drug-resistant to this antibiotic. We also confirm that the specificity and also accuracy can reach 95 or even 99% and above. And after building this machine learning algorithm, and after acquiring the strain, we don't need to do any other drug resistance testing or any other phenotype testing. We can get the genome data directly and we input it in our algorithm. It will return us a very accurate result. It illustrates the help from machine learning to our work. The third application of the machine learning, we can review the exchange of AMR between gorillas and humans in animal husbandry. In different times, we collected the samples of humans, environment, and animals, and we have the exchange of AMR, and we can predict the concrete results in the chicken farm. It is confirmed in our daily surveillance. It can predict very accurately. It is an ongoing project in our lab. We know that through MS and machine learning and for the vomiting gene of the Bacillus cereus, we can have good prediction. Now the ongoing project is about the C. fringens. We know that C. fringens has quite long cultivation time, and we have longer duration, and it is quite difficult to predict the strains and their features.
So currently we are considering to set the self-built database of the preferences. And this is very ideal for the accuracy. And they will complete the identification. So we can manage the identification of these preferences. So we can have this self-built database and to complete the detection and the identification. So currently we have more than 200 of these preferences. In the earlier stage, we have completed the very monolithic identification, and we have finished its WGS. So we can manage its phenotype and genotype results. Now in our library, all of its preferences have their types and the drug resistance and the other information in detail. So after the completion of this database, we would have very accurate data. If we want to carry out the identification of the other bacteria within a very short period of time, we would gain the very accurate database, and we would know whether this is a preferengen or not. And on this basis, we would also tell which type it is. If these types carry a specific characteristic, for example, whether it has specific drug resistance gene or the other genes, we would know. So this is our target. And currently we are still building this database, not finished yet. So I would like to share with you about this information here. As I have walked you through, my main topic today is rapid detection. WGS. Intuitively, you might find that WGS cannot be regarded as a real rapid detection technology. But from the perspective of the information, I do think that this belongs to the category of the WGS, because if you can manage this technique, then you can get massive data. And the volume of information is huge. So from this perspective, I do believe that this is a kind of whole genome sequencing technique. If we consider its development, the current WGS, from the first time that we carry out the WGS, it took about several months or even half a year to complete the sequencing. But now we can leverage this technology, and the sequencing in the early stage can be completed within several days. So this is really fast or rapid. We started to carry out this technology in 2013. As we know, at that time, the contamination scandal happened in New Zealand, the botulinum. On this basis, we carry out the response for this scandal. So we leverage the technology in dealing with this scandal, and we finish the whole genome sequencing and the other techniques as well. So we nailed the bacteria 1 week before the other agencies and institutes. So this is the best example case of the use of WGS. Also, we have done a lot, including our daily work and our scientific research. We have used lots of techniques in this regard. On the global stage, we reported the MCR 119 gene variant, Salmonella, and a novel MRSA strain carrying SCC MEC XV7A. So this is the first time on the global stage. We also use this technology in China to separate NDM1, MCR1, CTX M123 gene Salmonella. This is the first time of the separation for Salmonella strain in China, and lately we also found EMY MCR1 carried Salmonella. So this is also completed with the whole genome sequencing technique. In addition, we are also the first one in China to report the strains of the C. Utericum used in the food industry. So this strain can cause infant botulism, and the neonatal, the necrotizing enterocolitis. So this can be used as a probiotics for treating the diarrhea in clinics. In the generation of some chemical elements, this can also work very well, for example, in the production of some acid. So when we found this problem for these aspects, traditionally we believe that this bacteria is very safe. So we propose this concern whether this is really safe. We need to have further considerations for its safety. I think it's also helpful and necessary to introduce you about our analysis platform, because after we got the results, we need to do analysis. We should have the microbial background and the bio analytical background as well. So the platform would be helpful. With our hard work over the past several years, we have built this platform, and this includes the lethal bacteria and commercial industrial use bacteria, and the data of the other bacteria. So we have massive data in our library. Why we build this platform? This will be used as the database or library. In addition, we will also use this as a very helpful platform. So we would integrate the data from different sources, and this would be very conducive for the collection of the data to the analysis. So this covers the whole process in our work. We have also built a platform on the microbial resistance mechanism traceability. This will work for the identification and the traceability analysis for drug resistance. And this is capable of working as a one-stop platform. So what you need to do is very simple operation, and then you would enjoy the massive data generated by this platform. It's very efficient and very simple to operate. And this also is compliant with our routine mindset of microbial detection. And this has been leveraged on a very large scale as well. Currently, we have 36 institutions from 17 provinces. So they are all using our platform, and they have registered. And now we have about 500 on-site analysis. We have published this platform on our official website, and this is open nationwide. Due to stressful time, this concludes my presentation today with you. Thank you again. Thank you very much, Madam Dong, for your excellent sharing. Any questions from the floor? We got a question online. Predictive model on FML. Any outlook of application for the use of this predictive model of ML? I'm not sure whether I get your question correctly. The potential use of the predictive model of ML. Actually, we use the ML model for the feature or the characteristic that you desire, whether it carries certain characteristics of drug resistance or the pathogens or the other characteristics. And on the management of the pathogens, I'm not sure what aspect you're referring to. We got another question here, RTA false positive, how to avoid false positive. Currently, if you work for RTA, its main mindset is the clinical quantitative PCR. So in clinics, you would also suffer the false positive, so you need to strike a balance based on your purpose. If you aim to achieve to secure the accuracy, then possibly you would have a higher false positive. So you need to strike a balance. And it's very similar to the real use in the clinics. Thank you. Any other questions online or on site? The rapid detection of pathogens in lots of factories, plants, and the manufacturing sites. The detect indicators instead of pathogens. So any possible approaches to verify and deploy the rapid detection in the indicators. Companies now have lots of approaches. Sorry, so for these approaches, are these allowed by the regulation and law? From the state level, we need to refer to China's JB standard. Rapid detection. If you use it for your daily detection, then these rapid techniques are okay. But if you use it for the regulatory requirement, you need to refer to China's JB standards. Thank you. Sorry, I have a follow-up question. Your rapid detection techniques, the indicator includes the volume of bacteria and the other parameters. So China just issued the food safety regulation. They have included these key parameters, and I think you can refer to the latest version. Would it be feasible for us to have more new techniques of food detection? I think this is a requirement of lots of manufacturers. I think this will be the future direction of development. And we have used this approach in our daily work for a long time. But there is still a gap between our operation and the guidelines. Because first of all, in our daily operation, we are using this in real time. And secondly, this is becoming more matured. with the technological development. And as we have learned from the second speaker, the metagenome sequencing would be more matured in the future. So if we have an outlook into the future, this will be a very helpful alternative, because this is not limited to bacteria, also work for virus and fungus.
So I believe that with advancement of technology, China will consider more mature technology in the future. Thank you. Another thing, Ms. Dong, is that for the fast detection is used for the spoilage bacteria. No, because this technology is an emerging one in the past 2 years, we now only consider pathogenic bacteria or some pathogenic genes or other types of bacteria, for instance, spoilage or indicator bacteria. We still need some time to develop. Thank you. Any other question from online audience or from the offline audience? Based on our arrangement, we may have the 10-minute of Q&A session for each speaker. After each speaker finishes his or her presentation, so if you have further questions for the previous 3 presentations, you may propose your question. Any questions? No question from offline audience. No question either from online audience. So now we'll take a tea break and we'll come back later at 10:30, 30 minutes later. We will start the second half of the session. Thank you.
下午要跟你说一下。
我在考一个，我把它这个考出来，你那动电脑我动不了，我用不了。不是不行，我这考，我这动，然后里面也动。没变，我把它考出去3年了。
李凤琴老师，您能听到吗？李凤琴老师，
听得到。
李老师您好，咱们茶歇已经结束了，你这边可以。
So this concludes our tea break, and Madam Li, you can kick off now. We have our next speaker, Mr. Chen Wenyi, on site. So we will proceed our second half of the morning session. Our first speaker, Mr. Chen Wenyi, Merieux.
它的题目是下一代测序技术在食品质量安全方面的主要应用。陈博士呢
Nutrisciences Research Project Manager, share with us about key use of next generation sequencing, NGS, for improving food quality and safety. Graduated from Shanghai University in 2011, and then worked for Bright Diary. From 2016 till now, worked for Merieux Nutrisciences, Food Science Center, and mainly responsible for the microbial monitoring surveillance projects and food safety and quality.
His topic is a key use of next generation sequencing for improving food quality and safety. He got his doctoral degree of biochemical engineering in Shanghai University in June 2011, and continued to engage in postdoc research in state laboratory of dairy biotechnology, Dairy Research Institute, Bright Dairy, and Food Company. Now focus on foodborne diseases detection and traceability of foodborne pathogens, especially in milk and or dairy all the time. Please. Dr. Chen, are you online?
OK.
陈老师，你在线吗？喂。
喂，能听到吗？
听得到。陈老师，您请。
好的。谢谢两位主持人的介绍。
Thank you very much for your generous introduction. I'd also like to thank the organizer for giving me this opportunity. So today I will focus on this topic and share with you about the key applications of NGS in the field of food quality and safety. I'd like to thank my fellow speakers first for sharing with us about the applications of NGS. So in my bit, I will cover 3 aspects. First of all, the brief introduction to NGS, and then the key applications of NGS in food quality and safety. And last but not the least, I will share with you about when and how to use NGS when we come across the relevant barriers. So this is the first bit. We'll walk you through about the NGS technology. On the basis of the previous Gen 1 of this sequencing technology, we name this as a second gen. We know that the nucleic acid is a core of DNA. So we need to understand how to leverage this technology to get all genome information, the meta genome information. So this can contain massive data, and currently we are still in the process of WGS, so we can get the information of all microbial data. So this covers massive data, and secondly, lots of fermentation and food. If we want to discover the composition of the microbial components or the metagenome, we can use this technology. So I will cover two aspects to share with you. First of all, WGS, how we manage this. A brief introduction. The precondition for WGS is that we need to separate the pure the bacteria genome, and then we can use the second gen or third gen sequencing technique. Afterwards, we can get this sequence of the DNA. And then the strains of different types can be analyzed. And afterwards, we can get information of the relationship of these genes. So with this data, we would know the contamination sources, the time of contamination. On this basis, we can carry out detailed analysis to identify the sources, whether this is related to environment or the sources. So this is the concern of the enterprises. So we can use the WGS to deal with this. With this data, how can we have the preliminary analysis? So we can take different approaches. The first approach is that we can have WGS and then to analyze. First of all, we have software to have WGS comparison analysis or WGMLST. The different sources. After sequencing, we can compare the genes. After this approach, we will know their relationships. You can see the diagram on the right. We have 4 different isolates separated in different time. And after sequencing, we can use WGMLST for analysis. So we find that C and D are also related, which means that they have very close kinship. We can also leverage WGS data and to do WGSP analysis for single nucleotide polymorphism. And this is base by base approach. And then we can do the relatedness analysis by using the software. So you can see the sample 4 is closely or closest related to this sample. So with the analysis, we would know the relationship of the reference and our sample. This will be helpful for us to control in the follow-up approach. Briefly wrap up, WGS after sequencing and assembly, we would have the whole genome sequencing. So it has ADCG in its structure. On the strain level, we can carry out identification. And for WGS, we should collect more data, and this can be very helpful. Within a short period of time, we can finalize WGS within 1 week. And for the food manufacturers, this is very accurate and very effective for the tracing or traceability and identification, and also investigation. So later on, I will share with you about how to rapidly investigate and analyze the foodborne contamination. As you can see here, this is a chart from FDA and the Ministry of Agriculture in the US. So they have started to carry out the relevant sequencing years ago. And now they have more than 200,000 different strengths in their database, and they have the proprietary database. So we can leverage all this information in this database, have these in the WGS database, and then to have the comparison analysis, and then to gather more characteristics. So we can compare data in our database and to have positive information. This is another application of AGS. This is a meta barcoding analysis on 6S. Or meta barcoding. So after enrichment, we can get the data and to do an analysis from software. From the diagram, as you can see on the right, we would know the composition and the percentage of the microorganisms and whether there are changes as time goes by. So this is widely applied in the spoilage of the food. So briefly wrap up for this bit. Just for food industry or food manufacturers, implications mainly cover two aspects. First of all, WTS applications and the meta-genome analysis. So the first approach mainly targets our labs or units or our institutions. We get the genome and then to go through analysis, and then we can get the characteristics and relationship and features of this strain. On the other hand, the samples or the raw materials from the environment, we can gather samples, and then we can do this by the metagenome analysis. So this will be helpful for the quality control, surveillance, and the investigation of the contamination of the food and food safety. So approaches of these two different directions have been very frequent in the food manufacturers. Now I will walk you through the specific examples used to guarantee food safety and how WGS in food industry can play a role. So here, NGS mainly used in two different aspects. One is WGS, another is metagenomics. WGS is based on the pure sequence, and this is on the bacterial strain level. So currently the accuracy is highest and the resolution is the highest. On this basis, we can know its characterization, including resistance, and we can also do the relationship analysis, whether there are the relationships of the sources. And when we do traceability and investigation of outbreak or strain tracking or the other bio characteristics analysis, we can cover all this by the analysis of the bacterial strain. Also, this will be helpful for bacterial communities. It is very difficult for us to separate all the microorganisms, and some of them are not culturable, but they're very important in our samples. So in this regard, we have lots of challenges, but the metagenomics is a very helpful facilitation for these analysis. So we would understand the biodiversity or ecosystem diversity and the proportion of different elements, and if spoilage of food occur, then which elements play a key role, and also for the ingredients monitoring for the baby food, infant food. Raw materials have very stringent requirements for the baby food. So we can use the NGS technology for monitoring the raw materials to understand whether there are concerns of the contamination of the raw materials. Also, we can monitor the environment. what kind of microorganisms existing in the factory and whether they are relating to the spoilage, whether the environment is hygienic or not. And in the production of the food, if the environment is not hygienic, then the shelf life would be shrinked. So this will be helpful for extending the food shelf life. And also we can detect whether there are food fraud. I have covered two aspects about application. Now I will take WGS, especially its identification of the strain. For more clarifications, as we know that factories would get the pure strains, be it for the fungus or the bacteria. What we want to understand is the strains. So we can do the biochemical detection. We can do this on the genus or species level. But for the drug resistance and the other features and the relationship with different strains, for example, the raw material related strains and product related strains, the sources of the strain, how to better identify the sources. And we should do this through the sequencing of genes. And this will be helpful for us to tell the relationship with different strains. If we want to do this in a more in-depth level for the accurate traceability, what is the source of the contamination? In this regard, we need to do this accurately, so WGS would be helpful. There are two examples I will share with you. The first one is in 2018.
In 2018, and we had the largest outbreak of food poisoning in North Africa. And after eating the ham, and more than 1,000 people were poisoned. And after collecting the samples, and we isolated the pathogenic Listeria monocytal gene. Then we did analysis based on WGS, and we found which brand was poisonous and where the bacteria came from. Through which raw material, and we found the original contamination source. The second application is environmental surveillance. In 2017, there was an ice cream factory in the US, and it had the regular sample collection of environmental surveillance, and then the Listeria monocytal genes was isolated, and the data was uploaded in the database. Then the US authority found that the pathogenic information had a very high homogeneity with the genes from patients. So they consider that there was correlation between this strain from the factory with the patient. Then they collected a sample again in this factory, and they found indeed a pathogenic Listeria monocytal gene, which was very similar or with the genes from the patient. And they confirmed that the strain from the patients were from this ice cream factory. As a result, the factory was closed down for rectification. Then it resumed production. Based on WGS and its application in food industry, we mainly share with you that after the outbreak of food poisoning cases, the WGS contain enormous information for the accurate judgment. And then we can take precise control measures. The next part is about the 16S gene sequencing of diversity. And we mentioned that for the sample, when there was diverse microorganisms, and for the butter product, which is widely applied in dairy, for instance, milk and yogurt. In butter, the microorganism should grow slowly and there was no pathogenic effect. But after our sequencing, we found that for different samples, the microorganism mix is different, and there are also some conditional pathogenic bacteria. And for instance, there is Chronobacter strain from the butter. We thought it was safe, but actually there may be some bacteria which is pathogenic. When the butter is applied to ice cream or other dairy products, the bacteria may grow very fast. As a result, there may be some food poisoning incidents. Also, for the food factories, and there are different sections of production, and they may have cross interaction or cross contamination. And the source of such contamination is mainly people or tools. And for instance, for the risky section, including raw material warehouse, through the exchange of people, water, and air, or some errorless operation methods, there will be some contamination. Therefore, we need to have the full-scale screening of the factory. For some of the products which spoil earlier than the shelf life, we need to investigate the spoilage organisms and the origin of contamination. We need to have the full-scale investigation. And after investigation, for the susceptible root, we may have some sample taking. Then we may use the second-generation sequencing and analysis. Then we can make a judgment on the source of contamination, or through which material, raw material, and the organism enter the product. We know that food safety is the whole supply chain monitoring, and we not only need to guarantee the safety of raw material or the food factory. In each environment, there is a risk of microorganism contamination. We need to build a dynamic database of microorganism is quite important. From the paper in this page, for the community-wide monitoring, we need to detect the organisms and the potential pathogens. When the people, how the people flow in this community, we consider all these factors, then we can know the composite of the microorganisms and to make a judgment whether there will be the risk of food poisoning in the future. Then we can take some cautious measures beforehand. Through the whole supply chain concept, we can guarantee the food supply of food safety. We can not only limit our investigation in factories. For the spoilage bacteria or the microorganism database is important and essential for our precautious measures. If we don't know the composite of microorganisms locally and their potential risks, as long as we know the risk of potential microorganisms, we'll know that whether they will contaminate the food through certain pathways, then we can take measures accordingly. For instance, for one bacteria which contaminate the food through water and air, then as a result, the people will be contaminated and poisoned. For such spoilage bacteria database is important for the prevention and handling of public health emergencies. We can have the fast traceability, and we can have the comparison in the database, and we'll know what measures should be taken. Then we can quickly detect the source of contamination. In the end, we want to share with you that since WGS is quite advanced technology, so for companies, where can they apply such technology? For instance, in food spoilage, there are three types. One is microorganisms and chemical changes or physical changes. We mainly focus on the microorganism oriented food spoilage. There are some common phenomenon, for instance, food inflation, odor, or texture stratification, or soften. If it's microorganism, we need to find out the reason and in which pathway the food is contaminated and the food is spoiled. What is the root cause? Through what method we can find the source of contamination, so we ensure that in the future batches there will be no such issue. For the spoilage bacteria, we have several methods to deal with. To start with, we have WGS. If there is a spoilage in the food, we can speculate the cause. If it's bacteria, we should get the pure strain of the bacteria. Through sequencing, we know that whether the strain is pathogenic, and we can also compare with the previous database, and we'll find whether the strain is the cause of spoilage, and whether there is a correlation between this strain with the previous strains of bacteria. Then we can know about correlation, and we can know about the source. This kind of detection, the precondition is that we need to have the viable culture of the bacteria for the DNA sequencing. If the spoilage is caused by mold or yeast, we also need to get the pure culture of the microorganism. Then we can have the DNA sequencing, and through traditional stain method, there may be some shortfalls. For instance, the error rate is very high. So we need to do more. For instance, the sequencing method, we can have precise detection and traceability. Traditional detection methods have many shortfalls. Through the second generation sequencing, we can know about the real cause of spoilage and the features of the strains. Then we can take follow-up measures to control. No matter bacteria or fungi, we need to have the pure culture. Then we can have the NGS. For some samples, it is contaminated and spoiled, but it is very hard to isolate the microorganism. It is one common issue for companies. During isolation, the culture is not suitable for the growth of the strain. Then it's very hard for isolation. Then we can use NGS technology, and there is no isolation culture needed, and we just extract the genome. Then we can have the sensing. Then we use analysis software. As a result, we'll know the composite of microorganism in the sample and what's their percentage. So we can know the real cause of spoilage in the sample. Through this method, we can deal with the spoilage issue and find its root cause. In the end, we want to take this opportunity to introduce our company, Merieux Nutrisciences, can help you to deal with the spoilage and analyze the causes based on the metrics of the sample, and we can provide you with a solution, and we can also have the isolation and the verification. We also can help you with the WGS sequencing and also the traceability, so we can know the real source of the spoilage, as well as the homology analysis, so we can have the fast traceability. Apart from WGS application, we can also have the software for analysis. We have the one BA software, bionumerics, and we can have the effective analysis of the data from WGS. We can detect the homology and also the correlation between different strains. So this is the end of my sharing. Any question? Thank you. Thank you, Dr. Chen, for your sharing. Any question from our audience? In our daily work for Merieux, no matter the test strip and the identification equipment, and it is widely applied in our work. If you encounter some issues in operation and in the utilization of equipment, you may also communicate with Dr. Chen. If there is no further question, let's give a round of applause to Dr. Chen. Thank you. Dr. Chen, would you mind exit the full screen model? We may have some technical issue from the meeting organization committee, and just wait for a minute. Our next speaker is from the China National Institute for Food and Drug Control and Professor Cui Shenhui. The topic of his speech is overview of rapid detection technologies for food microorganisms. Professor Cui is famous in China in terms of the microorganism detection, and he is also the Deputy Director of the National Food Safety Standard Committee. So we'll welcome Professor Cui for the sharing. Thank you, Ms. Lee, for introduction. I want to introduce to you about the rapid detection technologies for food microorganisms.
The fellow experts have presented us very detailed. So in my part, I will cover the rapid detection techniques, approaches, and needs. So first of all, I'm sure that you all are very familiar about the food safety. And this is our significant concern. And with our analysis in the recent years, we found that about 200 illnesses relate to food safety, including the chronic diseases, and especially for the elderly and the children. And every year, about 76 million people in the US suffer from foodborne illness, and the most common intestinal infections are caused by viruses. So from these figures, it is apparent that across the world, food safety is a very significant issue. Also, you can have a very brief estimation about China's infectious population and fatality. Currently, we don't have a statistics officially, so you can do the very brief estimation on this basis. On this basis, for the detection, we can also take different approaches. From a long time ago, we started to carry out detection on the microorganisms, whether we can leverage the technology to detect and how to quantify. So we have lots of approaches and evolving as well. And this answers the question, what is it? What the bacteria is it? Is it Salmonella or E. coli? And traceability is important as well. What is the source of the contamination? More importantly, like the scandals and the lots of instances we heard from the other speakers, the traceability would be helpful for identifying the source of contamination. So these are the three main approaches. And now I will share with you about rapid detection indicator. A lot of the time we pursue for rapidness. This rapid regards the routine methods. And for the microorganisms, this is quite different to normal chemical and physical methods. On site, the chemical and the physics would be done within 30 minutes, But for microbial, this is more rapid. In certain environment, we can do ATP detection. We can get the very reliable results. Also, we have lots of monitors with the metabolism and the changes of the factors. We can detect the concentration of the microorganisms. So this is about indicator as well. And these are the technologies we can use for rapid detection. This is relatively very easy. The lethal microorganisms. So three main aspects would be important for us to answer. First of all, whether it has the salmonella or the other bacteria. And second, what is it? Exactly what is it? We should have the exact output. And what is the source of this microorganism? From the raw material or environment or any other sources. So traceability is important. So we have learned from my previous speaker, we can do this with the accurate sequencing. So this will be facilitated for us to identify the exact transmission approach. So the three main techniques I'll share with you today. First of all, theoretical analysis. This is based on DNA sequence specificity and protein lichen structure specificity as well. First of all, we should better understand reality. Among all the contamination incidents, microorganisms very frequently sent. We need to identify the source. This is come from water and then transmitted from the other medium and then into the food. So we can do the very brief analysis for this contamination. For example, in the chicken meat, salmonella in China, so in retail, the positivity would be close to 50%. So these 50%, 7.5 mpm per 100 grams, on average concentration, salmonella concentration. Well, for the VBNC, this is about 45 CFU per gram, and positivity is about 26.3%. And also for the Sakazaki contamination infant milk powder, this is about 0.3 mpm per 100 gram. So we cannot do direct detection. And don't be misled by the claims of the manufacturers. It is impossible to carry out a direct detection. These samples can be used in clinics. And this concentration figure is on average. So sometimes this is far lower than this average. And for the lethal pathogens, this is impossible for direct detection. And for the microbial detection, first of all, amplification would be necessary. It's imperative for us to complete this process. The enrichment, if any enterprise or any institution tell you that you can bypass enrichment, then they are liars. Enrichment cannot be bypassed. But this is a bottleneck of us. And for detection, we can amplify the microbial signal. What we frequently use is ELISA, PCR, and quantitative PCR. But every coin has two sides. It also has another side, which is contamination. So we should do amplification, but it also brings about contamination. So we should manage contamination. including the cross-contamination of the bacteria and capability of the detectors and the environmental contamination as well. So in the detection, this is our basic theoretical principle or rationale. Let's come back to the process of detection. This is a very classical routine for food detection. So we should do sampling from 25 grams per milliliter. If this is salmonella, we can do the pre-enrichment for salmonella first, and then selective salmonella, and then selective plate separation and staining, biochemical identification, and serotyping. This is a very typical process for the pathogens. As you can see here, we are amplifying our targets from 25 grams to the pre-enrichment. This is the amplification, and then to the next step, another amplification. So we do amplifications step by step, and then the identification and the traceability. So after the first step and second step of signal amplification, we can carry out detection. This is formulated by China's GB standard. Why we should detect to 25 grams? Because this has been amplified by 2 steps of the signals. So in the microbial detection, first of all, it's impossible to carry out on-site rapid detection for food microbial, like within 2 hours. This is impossible for the foodborne pathogen. And second, we embrace the rapid techniques. You can refer to the different information, or you need to select a baseline. Our baseline or a reference is China's GB standard, the national standard. Identification compared with the physiochemical and traceability as well. Chemical and physical, not accurate, but microbial, more accurate. So these 2 areas are different. Normally, after pre-enrichment, we have 1 ml for sampling, and the selective enrichment, we have 10 ml for sampling, and then to go through the next stage. In the pre-enrichment or selective enrichment detection, sensitivity requirement is about 1,000 CFU per ml. So in the pre-enrichment, you can set a detection. Or if not that sensitive, or your medium is not that good, then in the second step, you can do detection. With this analysis, I want to share with you that normally we should do the overnight culture, about 24 hours for the so-called rapid detection. Normally, this will be completed within 72 hours to complete the whole process. If you have the high sensitivity detection method, which means that it can reach 1,000 CFU per ml, then the site of detection can be put forward to the step after pre-enrichment, or to replace the selective enrichment. You can have the multi-target high sensitivity method and AMP method, or the multiple fluorescent quantitative PCR. We have done lots of systemic analysis in our labs. Sensitivity is close to PCR, which means that if the target is very low, then this will not work very well without enrichment. The concentration of pathogens in the environment will be very hard to reach 10 to 3. In this process, you also need to control contamination. This is a example case of enrichment, you can have a detection for 3 or more pathogens. In the same sample, you can select environment for enrichment and then to do detection. And you can use biosensors. Several years ago, some of the colleagues also do experiments in this way. They can identify with the antibody and to generate fluorescent signals to detect whether there are microbials. This is automated ELISA. Also work for detection of the microorganisms. So these are for bacteria, but for viruses, currently we don't have the ideal methods, especially noroviruses. You cannot do enrichment. So if you want to detect these viruses, you can do this in a very limited categories or types. The DNA amplification and immuno detections. In addition to PCR and immuno, we don't have the remarkable breakthroughs. So these are the approaches for the detection. First of all, identification. In our conventional JB standard, staining and the biochemical would be used for identification. Very conventional, very traditional. This was established in 1920, including the gram staining. And now we have the polysaccharide and protein and DNA identification. Compared with the serological, their accuracy level are not the same, more accurate. So for the conventional biochemical and immuno and specific genes and DNA sequencing and the MS technologies, we all use them for identification, especially MS technology.
All these technologies and their speed can reach the detection of one microorganisms in several minutes. For DNA amplification and sequencing, it is done still within an hour. For the identification is in the genus or species level, whether it is Salmonella or E. coli, and we identify the microorganisms on this level, not on individual level. If you want to complete the genus or species identification, means that we already have the pure microorganisms. So the method requirement is not very high. If we have 10,000 or 1 million CFU, the sensitivity requirement is low, but it needs to be fast and precise, and needs to cover the level of genus and the species. We need to cover all the serotypes of Salmonella to have the precise identification. We also need to have the requirement for multi-pathogens. For instance, the pathogenic E. coli, and for the 5 types of E. coli, we need to have the precise identification. Sometimes we also need to have the anti-cross contamination capability. Sometimes the culture is not pure. For instance, PCR method is very good. It can deal with the cross contamination. For MS, the result may be not precise. For some slow method, for instance, PCR, it has its own advantage. For some fast technology, for instance, MS, it is not so resistant to cross-contamination. Some examples on the DNA sequencing for high specificity, for instance, fluorescence, quantitative PCR, and the LAMP method. It has identification of the genus or species level identification, or have the sequencing of the gene, for instance, cysteine as RNA sequencing. It is on the genus level, but on the species level, the result may not be so precise. It's more a comparison of the sequencing. For cysteine as method on the genus level, it is accurate. We also have the GI-CI method. colloidal gold immunochromatography assay, which can be done in several minutes for the antigen and antibody reaction. For some of the method is even more quickly than the chemical physical method. We cannot confuse all these methods. Sometimes it is very hard to explain it to government officials because they require some like dumb method and result within 3 minutes. But in the lab, we can do that, but in the normal environment, it's very hard. For the body TOF MS, which is also very fast. We're using MS, we can have the different images, then we can compare with a database, and we can have a quick identification of bacteria, which can be done in 3 or 5 minutes. So I have introduced some of the fast detection method of microorganisms, or some of the most fast and the fastest and the most reliable method is MS. And also another important thing is about traceability of microorganisms. Traditional traceability is the serotype technology. It is the most ancient technology. It is still preserved. A traditional method for the national standard of detection, the serotype method is still maintained. We need to have the accurate genus or species result. For some of antibody may have cross-contamination with other species or genus. So we need to have the accurate result, and then we can have the traceability. Or the traceability is more about species based. If they come from different species, it is not meaningful to have the traceability. For the traceability method, the most ancient one is the serotype. Now we have some new method like PFGE, like the flow cell, sequencing, etc. We have the timeline. Since 1920s, serotyping was proposed. Then we have the phage typing. Then we have the plasmid profiles and ribotyping in 1980s. Then after 2000, we have the PFGE, MLST. In 2015, we have the whole genome sequencing. Currently, PFGE, especially PFGE, has been phased out in overseas. Or RFP, which is quite simple and is still adopted in some farmers. But now we are moving towards the whole genome sequencing. For traceability, this is a trend. We can get the largest amount of data. For traceability, for microorganisms, from this image, we go through the process of improving understanding. Initially, we only get the serotyping, only some antigens. It's like the blind man tried to figure out the entire shape of an elephant. Initially, he only touches the leg of the elephant. So he needs to improve his understanding gradually. If we violate the overall sampling method, if you're interested, you may observe the development history of microorganisms. And there are different types of traceability methods, but only a few of them are preserved. For instance, the ribosome, the ribotyping sampling based on the cysteine as gene and its distribution in the ribosome. And we also need to figure out the difference of distribution of genes and also the loci. For MST, for PFG, we may just find different number of loci on gene. To some extent, they're reliable, but after 2010, the emergence of whole genome sequencing, initially the cost was very high, but now it's much lower. Now, under cheap prices, we can have the WGS, and we can get the image, we can get the passage of the bacteria. Currently, the sampling cost is very close to the overall cost of sequencing, then is it still necessary to have the sampling, or we just have the sequencing of the whole population? For some of the mature website, one is from the Denmark, and after uploading the data of genome sequencing, after PFGE, and they can make a judgment on the serotype or on the MLST or the plasmid, as well as individual independent genes and Nile genes, and they can tell all this information. If the contact is relatively long, you can get the genetic structure. For different strains, we can have the traceability analysis because we get overall data. After introduction from expert, from metrics, and we can build the precautious method, the precautious system. For instance, the microorganisms in equipment, in the environment, in the workflow. You can have the WGS. Then you can build the database based on that. After updating the database regularly, you can make the control and analysis of contamination level in the working flow. Based on such precautious system, you can make a judgment whether there are loopholes in your management and operation. For some of the food companies, the control is like between 0 to 1. If it's 0, it's safe, and if it's 1, or if it's 1 is safe, if it's 0, the factory needs to be closed down. You have only two choices, and positive or negative. Then the company needs to find a compromise in between. For instance, we can start from 0 to 1, then we expand it to 0 to 100. We find some relevant data, we can have the defense system. It means that if we are lower than 60, we can have the defense measures. If it's lower than 60, we just close down ourselves and without the interference of the authority. To sum up, for the microorganism detection, we have some reliable method for fast detection. The principles and methods are different from traditional culture. It is mainly based on the protein, DNA. In 24 hours, we may complete the detection for some bacteria. We also can have some fast detection in the core, and we also need to have some serotyping and faster traceability method. Thank you. Thank you, Professor Cui, for your excellent sharing. Any question for Professor Cui maybe related to his speech or anything you encounter, any problems you encounter in your work? You may propose it to Professor Cui. So any questions? For our offline audience, any questions? No questions. Then let's give a round of applause to Professor Cui for his sharing. Thank you, Professor Cui. We want to take this opportunity to introduce our next speaker. She is the Managing Director, Liao Yongwei from Romer Lab APAC, Singapore. Her topic of speech is Cytoquant bringing food safety and hygiene to the next level through the world's first mobile flow cytometer. Ms. Liao has the bachelor degree in California University, and she also has a certificate from the Cornell University in food safety. She has rich experience in food safety, no matter for the meat and allergen, and now she is a managing director of Romer Lab APAC.
Holds a Master of Business Administration from the California State University, East Bay, USA, and received the certificate of attendance from the Conagra University Food Science Dairy Training Program in 2013. And now she works at Rome Labs. Ms. Liu, can you hear me?现场听见我声音了吗？
可以听得到。
哎，
Yes, we can hear your voice.
廖女士，她是在线下还是在线上？
李老师，那个永卫他这边是提供的视频。
Ms. Liao has provided us with a video speech, and we will broadcast the video from offline.
视频吧，好吧，谢谢。
好的，好的。
We are glad to share an update on Roma Labs' latest new product that will have launched in the market. The name of the product is called CytoQuant, and it will bring food safety and hygiene to the next level, as this is the world's first mobile flow cytometry to help in our food industry. Today, I will share a little bit on the impedance flow cytometry and where this basic technology actually evolved into food production today, and also discuss about the sensitivity, accuracy, and the advantage for application in the food industry. And here it is how the CytoQuant actually looks like. We have a meter and also a swab. We call it the swabbing kit could be inserted to the reader. Let us take a look at the test systems available in the market for surface microbiology analysis. On the left-hand side, you see the direct methods such as plate count, which we know from time to results perspective, it will be available in 2 to 5 days. And for microbial count, accuracy, and robustness, direct methods such as plate count are not influenced by chemicals and surroundings. Whereas in the indirect method like ATP assays, time to result usually much more rapid, less than 30 seconds. Accuracy-wise, take note, it measures biological residues but not bacterial, and the robustness itself is heavily influenced by chemicals and surrounding. And for CytoQuant, we consider it as direct method. Our time to result can be done in 30 seconds, and the accuracy itself, it measures 2 readings with the bacterial count as well as the particles count. And take note, the robustness itself is actually not influenced by chemicals and surroundings. We actually did a lot of research behind the scene to develop the technology, and basically cytometry in this measurement actually help us to identify the cell characteristic, and it is able to measure the cell characteristic itself. And with this technology, it's also based on impedance. It's actually an electrical resistance that we use an alternating current to actually understand the characteristic of the cell behavior. In summary, flow cytometry impedance introduced by particles and cells passing through a microfluidic cell. In our technology platform for cytokine, the impedance actually relies on the frequency of the electrical current that we send through. And multi-frequency measurements of particles actually offers detailed structured information. With this, we'd like to explain that the bacterial structure being unique actually differs from other particles. And because of this uniqueness, we're able to count the bacterial. And this counting actually is based on the cell wall or the cytoplasm. In other words, today with this technology, we're able to count the number of live intact cell bacteria. And let us explain how this works. In a normal situation, when cytokine from detection principle itself, conductive and non-conductive particles at low and high frequency actually behave differently. For a conductive particle, of course, the conductivity will pass through, whether it's low or high frequency. And for non-conductive particle, whether low or high frequency, it will create an impedance around the particle itself. And if you compare this technology for bacterial, it actually have a different phenomenal behavior. The bacteria at two different frequencies, both low and high, give us a different signal. When we do these different frequencies, on low frequency, the bacteria actually have an impedance around the particle or around the bacterial cell wall. But when we have high frequency applied through bacterial, it will provide a flow through the cell itself. So with this different frequencies of behavior, we're able to detect whether the bacteria is alive and we can actually count them physically. And with this, the summary of the detection principle summarized in this chart, conductivity both in high and low particle, this differential behavior for positive and negative in the bacteria-wise for high and low frequency, we were able to detect it through a pulse calculation. And that is how the cytometry works for our cytokine by RomaLab. And what do you detect with impedance cytometry? In this case, for particles, could be also residues, and it also detect intact cell or intact bacterial cell, and we call that IC, intact cell wall or intact cell, up to a defined diameter of 10 micron. And we actually can do this detection irrespective of special growth requirements such as whether the bacteria is aerobic or anaerobic, and whether it's a pH high or low, whether the nutrition is of high fat content, whether the salt concentration of the food itself is whether high, or whether the temperature in the surrounding is actually different, whether there's high temperature or cold temperature. And it is regardless or irrespective of any lag time. And we can do this impedance cytometry even though we have no incubation time required. And best of all, impedance cytometry is able to detect viable but non-culturable bacteria. And this is a really real advantage for food manufacturers. And whether it's a solid or liquid material or media, we could cover the entire range. And actually, to put it in perspective, there is a very important background to currently what we know about bacteria. Of the population that we're aware or could actually culture today with our technology in media culture, we have 1% of existing bacteria that are culturable. And this is the reason why results currently, if you deploy the flow cytometry or impedance cytometry, deviation from plate count usually you'll observe, because for mixed cultures like production surface samples, we'll be able to see a difference in the result. And probably because of this 1% of existing bacteria that are culturable, if you do a comparison. On particle counts, so particles actually represent any residues not classified as bacterial. Particle count is an indication of cleaning efficiency, but it doesn't say anything about this infection. So in this impedance flow cytometry that we have developed for cytokine, we have actually have an upper limit of quantification and also a lower limit of quantification. Take note, particle counts usually are less reproducible with our cytokine, actually because the consumable themselves could actually emit the particles as well. Here, we'd like to share a current application for flow cytometry. It's typically used in drinking water analysis, and possibly a system that's called cytoflex is also frequently being used. A little bit more on the accuracy and sensitivity of our cytokine. We have the time to result at 30 seconds, and for the lower limit of quantification for intact cell live bacterial, our limit of quantification, the lower limit is at 15,000 cells per ml, and also for particles at 50,000 particles per ml. And take note, this is not influenced by chemicals and surroundings. Here in the cytokine workflow is actually quite simple. You open up the swabbing kit, followed by swabbing the surface to collect the bacterial, followed by suspending it with shaking, and return the swabbing kit into the cytometer itself. So when you press OK, there will be a 30 seconds for the results to come out, and you'll see on the right-hand side here, intact cell reading per ml, as well as the particles per ml. In this slide, I would like to share the comparisons lab study that was done for the CIFSQ colony forming unit versus our intact cell by cytokine. We actually did a PSA plate for CIFSQ per ml for two of the pure culture Staph aureus ATCC 25923 as well as E. coli ATCC 25922 pure cultures when we spike on the surface, and we did a similar recovery for using the cytokine in terms of intact cell per ml. Here the result can be shown in the R square for both is actually above 0.995. And as you can conclude from this chart, that both in terms of linearity as well as accuracy, cytokine intact cell reading is actually as accurate as the CIFSQ forming TSA plate unit. And here in the comparison laboratory study 2, we actually extended a lot more pure culture on the surface to make a comparison between cytokine and the plate count. Here we extended including the Salmonella enterotica, Pseudomonas aeruginosa, as well as Enterococcus facilis. And the results here shows that both the intact cell from cytokine and the CIFSQ reading is actually very equivalent. With this, we'd like to conclude from laboratory studies 1 and 2. Cytokine generates slightly higher counts on the bar chart than the plate methods, as not all cells form colonies, and some colonies emerge from more than one cell. The physiological state of the bacterial strain strongly influences its ability to form colonies. Therefore, plating the same strain with a similar cell count at different times can result in different plate count as well. And take note, dilutions of the same culture sample generate correlating cytokine results at a given time. In the comparison laboratory study 3 that we are actually going to extend further, we actually did a comparison study between different bacterial counting methods that was conducted by the Karl Landsteiner University of Health Sciences in Krems, Austria. The method that we use to make the comparison with cytokine includes the fluorescent microscope method, fluorescence flow cytometer method, which in this case we have the brand name called CytoFlex, as well as our impedance flow cytometry cytokine technology, and last but not least, the aerobic plate count, we call it the TSA plate. The goal was to verify in this laboratory study 3 and to compare the intact cell counts of the cytokine mobile flow cytometer with all the other methods. Here's the results. Comparison laboratory study 3 shows that between the microscope fluorescence method as compared with cytokine impedance flow cytometer method, they are almost equivalent. And cytoflex actually return a much lesser reading compared to the fluorescent microscope method as well as cytokine. And in fact, the plate count, as predicted, TSA plate return the lowest count for the bacteria itself. We would like to summarize in the comparison laboratory study 3 here with a table. And we included the standard deviation as well. And here you can see for E. coli ATCC 25922, the results from cytokine as an impedance flow cytometry, the standard deviation at 9% is among the lowest compared to the rest of the method. And for Sulfurous ATCC 25923, the results in the standard deviation is also 6%, the lowest or one of the lowest compared to the rest of the method. And in conclusion, from the laboratory study 3, cytokine shows results very close to that of the reference microscope method. Cytokine showed the lowest average standard deviations of all methods. As expected, plate returned the lowest bacterial count. The fluorescence flow cytometer, in this case cytoflex, has significantly lower bacterial count than cytokine as well as the reference microscope method. From the picture that you can see here, the portability and convenience for food production operator to actually bring this flow cytometry cytokine into the production floor, it is really very easy. It is a handheld device weighing less than 742 grams, and the size itself is about 230 multiplied by 80 times 95 millimeter. Operating temperature is at 5 to 30 degrees Celsius, and is actually chargeable with battery operation. Counts, and it allows us to actually have a reading of intact cell count as well as total particle count in 30 seconds. With this technology available in the market right now, we have find many wide applications for cytokine, as it is the only first world mobile flow cytometer able to help in biofilm detections, and is also able to detect microbial load on the eggshells. This is for brand protections, and also for the rinses water testing applications, as we are all familiar of here. And definitely a huge part of these applications for cytokine will be on the surface hygiene control and risk assessment. We also found a very unique positioning that cytokine is able to help to understand the biotic life culture that is used in dairy as well as used in the aquaculture industry, whereby it can learn whether the probiotics bacteria is growing well. And the quantification is done so easy within 30 seconds. Here we'd like to share the cytokine applications on the largest egg producers in the industry. Here we can see that the grandparent egg is highly costly and valuable in this industry. So measuring the bacterial loading on the eggshell will indicate whether the contamination is so high and control if it's in place. So typical measurements for bacterial loading will also be extended to egg belt, transportation trolleys, hatchery egg beds, as well as hatchery chick conveyor belt. The key part that we want to mention here is that we have hatchery total contamination with microbial load that is so high on the eggshell, and it actually may affect the egg itself going into incubation and explode, and we call that adventurous eggs. To avoid this adventurous egg phenomenon, the best way is to actually have a very good hygiene standard for the entire egg production. So to give a good indication of the microbial load, using cytokine will be very meaningful in risk mitigation to avoid adventurous egg. And next, we'd like to share the largest applications in poultry as well, whereby the challenge in Campylobacter and Salmonella in terms of sanitation verification with rapid cytokine is actually feasible. Why? It started from the production on the right-hand side, whereby you can see the chicken carcasses with the antimicrobial treatment being done, and each of the grey color or darker color shade is actually indicating the sampling sites whereby microbial analysis need to be done. Because of the cytokine rapid results availability within 30 seconds, this could be a very good advantage whereby we can access the microbial understanding in terms of bacterial count, intact cell count, to understand which step is actually having a high bacterial load. With this, you can use the information to actually help in sanitation verification, whereby cleanup can be done almost right after the 30 seconds if the microbial bacterial intact cell count is too high from cytokine. With this, you can actually be assured that Campylobacter and Salmonella could be avoided. And using a very good method in sanitation verification to understand the disinfectant capability and capacity, this will really help to mitigate the risk far in advance. And next, I'd like to share a little bit more also on the surface hygiene, whereby we find very interesting viral but non-culturable bacterial phenomena or incidents. In the largest poultry producers, we are also very worried of Campylobacter. And one of the opportunities cytokine could actually be applied is actually in the detections and quantification of viable but non-culturable Campylobacter jejuni. In the current regulatory requirements, if we have a system as rapid as 30 seconds from cytokine, we could actually help the regulatory to actually go for detections within 4 days. Because of the requirement in rapid reporting of Campylobacter in certain regions in Asia, classical method and traditional method is actually too slow. And deploying cytokine will actually enhance the regulatory speed in responding to issues like Campylobacter outbreak. And how does it work? We'd like to share a little bit more as well on the other applications that we're seeing cytokine is very feasible and effective. That is on the probiotic culture applications. The use of probiotics in shrimp farming, particularly in Indonesia, has been a very important driver for the industry. In fact, it is known as an alternative method to not use antibiotics. With the increased safety and sustainability for the environment in Indonesian aquaculture, we are able to bring cytokine for the application and measuring the number of bacteria contained after activation. An example here is actually done in Indonesia. Two different probiotic lot is actually able within 30 seconds after preparation, we are able to tell the number of intact cell bacteria before we actually feed the probiotic bacteria to the shrimp farm itself. So you can see how effective cytokine is for the monitoring and control of actual bacterial intake for probiotic into the shrimp itself. For human consumption, we also have live probiotics. And here is a study done in New Zealand that we have done for probiotics, whereby different dilutions of the probiotic was being prepared, and we actually have the cytokine measure the intact cell or live probiotic count. You can see the reading here that cytokine is able to tell each dilution factors very easily within 30 seconds. And with this rapid capability, you can actually monitor the live probiotic culture production online. Earlier, we have seen all the different applications of cytokine. Here, I would like to share a little bit more on the exciting application that can use cytokine for biofilm detection. Earlier, we shared flow cytometry detects bacteria and is a direct cell counting technology. Cytokine is a flow cytometer that does this in 30 seconds. Biofilms are formed by bacteria. In other words, cytokine with the flow cytometry technology will still be able to detect biofilm. Attached is an interesting scientific paper about the topic, also naming explicit flow cytometry as possible solution for detection. I have come to the end of my presentation. Thank you everyone for participation today. We hope that you learned a lot more about cytokine by NormAccurate, bringing food safety and hygiene to the next level. And this is actually our world first mobile flow cytometer, cytokine. Thank you very much.
Distinguished representatives and delegates, and since Professor Lee has some emergency to deal with and she's not online now, so I will represent the moderator to summarize our session. Since Ms. Liao is our last speaker, and to start with, we are very grateful for everybody. In such circumstances, and you are still present in the meeting, it means that food safety is the focus of the industry. Thank you very much. So this concludes our session B. We are going to the lunch break, and our next session will start at 2 p.m. Thank you.